So we've been talking all throughout the course about the various structures of the body and the functions that those structures have. Those structures are made by molecules that are put together by enzymes. Enzymes are proteins that are encoded by genes. So our structures and our physiology, the anatomy of the body, all of our traits are ultimately determined by the genes that we have. So in this final chapter of the semester, we're going to talk about how the traits that we have are inherited. Our understanding of genetics began with Mendel and his garden peas. He was studying traits that were either all or none. So he got lucky that he had simple patterns of inheritance that he was able to track with his garden peas through multiple generations. Human traits are more complex. Many of our traits are combinations of multiple genes, which makes them more difficult to study. So knowing the sequence of the human genome, that is the entirety of the DNA contained in the nucleus, has been a goal of modern research in genetics. And the Human Genome Project was designed to do just that, decode the DNA of the human genome. It's a multi-billion-dollar project that took many years and multiple labs to complete. And now we have a fairly good understanding of the basic sequence of the human genome. The genome is the entirety of the DNA contained in a cell. So it's all the genetic instructions for making all the proteins that that cell would need. We have two sets of each gene, one from our mom and one from our dad. So that means that we have one of each chromosome from mom and dad. There are 23 pairs of chromosomes in the human genome. 23 pairs of chromosome. One of that pair came from mom, the other one of the pair came from dad. So we have a total of 46 chromosomes in the so-called diploid cell. That is all the cells of the body except for the gametes. The egg and the sperm are the only exception to this, or the cells that immediately precede the egg and sperm. All the other cells of the body are diploid, which means they have 2 of each chromosome for a total of 46. Again, there are 23 pairs. The first 22 are considered autosomes. They don't contribute to the gender of the individual. And then the 23rd pair are the so-called sex chromosomes, and they do determine the gender. So they're either X or Y. Those are the two alternatives for the sex chromosomes. And if you have an X, if you have a double X, you are a female. If you have X and Y, you are male. So one of these came from mom, and the other one came from dad. This one came from mom, and this one came from dad, because dad is the only one that can contribute a Y chromosome. The chromosomes an individual has can be displayed in what's known as a karyotype, where the chromosomes have been dyed with some fluorescent dyes that are going to light up under a microscope, and they've been laid out using a computer so that each chromosome, each pair of chromosomes are next to one another. These are homologs. So this is 2 examples of chromosome number 1. One of these came from mom, and the other one came from dad, and they're known as homologs. And then chromosome 2, same thing, 3, 4, 5, on up to 22. And so these first 22 are known as autosomes. These do not encode for traits associated with gender, and they go from chromosome 1 to 2, 3, 4, 5, like I said, in decreasing size, pretty much, more or less, is the idea anyhow. And then the final 23rd pair are the sex chromosomes, where the X is going to be larger than the Y, even though they don't look like X's and Y's right here. The X is larger than the Y is quite small by comparison, so this apparently is 2 X's, which means that this is a female's karyotype. Since there are 2 homologs, or 2 copies of each chromosome, one from mom and one from dad, that means there are 2 of each gene. The genes are encoded on the same location of each chromosome, known as a locus. That's the location on the chromosome where you can find that particular gene. You'll find the same gene in that location on the maternal and the paternal chromosomes. But you might find slightly different variations of the gene. So the gene might code for eye color or something like that. You might find that same eye color gene on mom's chromosome in the same location on dad's chromosome. But they could be coding for different colors. So the different genes that are found in each location or locus are known as alleles. These are variations of the same gene. You might get the same allele from mom and from dad, in which case you would be homozygous for that particular gene. That is, the locus on each chromosome, you would have the exact same DNA sequence. If the DNA sequence is different at those loci or locations, then you would be heterozygous for that gene. You would have two different copies of the genes or two different alleles, heterozygous. So if an individual is heterozygous for a particular gene, they've got two copies or two different alleles of that gene, usually one of those alleles is going to be dominant over the other. Its expression, the trait, the expression of the trait that that gene codes for is going to be expressed over the recessive allele. So if we're talking about eye color, for instance, brown eyes is dominant over blue eyes. And when we talk about genes, we oftentimes abbreviate the gene. We give them a letter B to denote eye color, for instance. Where capital B is the dominant allele, and the lowercase b would be the recessive allele, the one that doesn't get expressed. And same holds for any other gene. You give it a letter, and the capital letter is going to be dominant, the lowercase letter is going to be recessive. So in the case of eye color, B is a letter, capital B is dominant, and it means that the eyes are going to be brown. Blue is a recessive trait, and it's a lowercase b. So if you have one copy of the dominant allele, then you will express that dominant trait. In this case, eye color, if you have one copy of the brown eye allele, then that individual is going to have brown eyes. So there are two different combinations. You could have a dominant from your mom and a dominant from your dad, or you could have a dominant from one parent and a recessive from the other parent, in which case, in both of these examples, you would express the brown eye trait. The dominant trait comes through. So there's two different ways, two different genotypes that you can get that same trait expressed. Whereas a recessive trait is only expressed, or it's only observable, or its effects are observed, if you have two copies of that particular recessive allele. So in the case of blue eyes, we're talking about two lowercase b's. You got one recessive from your mom, one recessive from your dad, and this individual would have blue eyes. So those letters, combinations of letters, display what's known as the genotype. In our example of eye color, there are three different genotypes that are possible. Got two alleles, lowercase and uppercase, or I should say the uppercase and lowercase, I guess. So three combinations, how you can arrange those from mom and dad. You can have both uppercase, both lowercase, or one of each. These are distinct genotypes, the patterns of alleles that you have for that particular gene. The phenotype is what is actually expressed physically. So in our eye color example, the phenotype is either blue eyes or green eyes. So in the case of eye color, there are two different genotypes that give the same phenotypes. Big B, big B, big B, little b both lead to the brown eye phenotype because, again, the brown eye trait is dominant over blue eye. The other phenotype possible in this example is blue eyes. There's only one genotype, little b, that's going to give that. So a homozygous recessive will have the blue eye trait. Each of us has a unique genotype and phenotype, because of several things that occur during meiosis or fertilization. Independent assortment of chromosomes, crossing over, and random fertilization that occurs when any one of the given sperm that might make its way in to the female finds an egg. In meiosis 1, there's a couple of really important things that are going on that are going to lead to some diversity, genetic variation. There's segregation. Remember, there's 2 alleles of each gene, or each trait. One came from mom, one came from dad, and during meiosis 1, those can get separated. Those alleles can get separated to 2 different daughter cells. So, eye color example, one of the daughter cells in meiosis 1 is going to get, say, if you have a heterozygote for eye color, one of the gametes formed in meiosis 1 is going to get the dominant trait, the other one will get the recessive trait. That's known as segregation. We're separating the 2 alleles in meiosis 1. And then there's this concept known as independent assortment, where now we're considering alleles of different traits and how they're going to separate out during meiosis 1. So for instance, maybe we're talking about hair color. What the frick? Alright, I don't know why we changed our example all of a sudden from eye color to hair color. I assume this is supposed to be eye color. B for eye color. Right, we're talking about one trait, eye color, and another trait, say, the flexibility of your thumb joint. Whether you have a loose thumb joint or tight ligaments in your thumb joint, the so-called double joint trait. These traits, eye color and the thumb joint trait, are on different chromosomes. So when they're segregating during meiosis 1, those alleles are going to segregate completely independently of one another because they're on different chromosomes. So when you consider the 2 together, there are different possibilities of inheritance shown here. You have a heterozygote for eye color and a heterozygote for the thumb joint trait. We can have dominant, 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 recessive, 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 dominant, or recessive, recessive. Many different possibilities here. And there are many, many different genes, many different alleles on the 23 different chromosomes that can all, more or less, assort independently of one another. So this leads to a tremendous amount of genetic diversity. Since you have 23 chromosomes and you have 2 of each chromosome, the possibilities are 2 to the n, where n is the number of chromosomes. And that leads to a tremendous number of genetic variations. And we're just talking here about what's going on in meiosis 1. So segregation and independent assortment depend on how chromosomes line up at the metaphase 1 plate. Remember the tetrads here. You got the 2 sister chromatids on one homolog and 2 sister chromatids on the other homolog. And you got 2 homologs that are physically connected at metaphase 1, and they can line up in any different number of patterns. We consider maternal and paternal. So, for instance, all the paternal could be on one side, and they're going to go to the same gamete, and all the maternal could be on the other side and end up in the same gametes. Or you could have a mix of maternal and paternal, many different combinations. And this example is just showing 3 different chromosomes. So we have 23, so there's a tremendous number of gametes that can arise from meiosis 1. And another factor that's going to lead to a lot of genetic diversity is what happens during crossover, which involves genetic recombination. When the tetrads form and the 2 homologous chromosomes are brought together so that they can line up at metaphase 1, there is this actual crossing over. It forms a chiasma. And in the crossover, part of the DNA of one chromosome gets threaded into the other chromosome and vice versa. So there's an exchange of genetic material between the chromosomes during recombination. And these chromosomes are now considered to be recombinant. They're new combinations of chromosomes. So you mixed up the maternal and paternal chromosomes so that they're no longer just maternal and paternal. Now they're a mix of each, and this greatly increases the amount of genetic variation, because in any given chromosome there could be multiple crossovers up and down the length of the chromosome. And again, these crossovers are required in order to bring the tetrads together at the metaphase 1 plate. So here's a couple of homologs to consider. And we're looking at two different traits, even though there's multiple genes up and down the length of each chromosome. There's thousands of genes, but let's just consider two. They changed nomenclature on us again from B. Now eye color is an E. And we're also looking at hair color. These are both on the same traits on the same chromosome, where the green is maternal and the blue is paternal. In crossing over, one of the non-sister chromatids, this green non-sister chromatid, exchanges material with that blue non-sister chromatid. So there's an exchange between the homologs here. Remember, sister chromatids are the same color. Non-sister chromatids are different colors. Eventually the chiasma, or the crossovers, are going to break to allow the chromosomes to separate when they're supposed to. And that's going to leave new combinations of chromosomes. One of these chromatids is the same as it was before, but here's a new recombinant chromatid. Same thing here. This is a new combination of genes here. So we created a new version of that chromosome. Now when meiosis is complete, we've got 4 different versions of this chromosome now. And again, this is just showing one chromosome and 2 different alleles, or I should say 2 different genes here, and the different combinations possible there. But again, this is happening all up and down the length of each chromosome, and there's 23 chromosomes, so a tremendous amount of diversity can be generated during meiosis with the crossing over. And then of the gametes that are formed, any number of those can potentially fertilize the gamete from the other sex. So when we're talking about the different number of eggs that could be produced in meiosis and different number of sperm that could be produced, just a tremendous amount of diversity possible with each gametogenesis program. And then when you bring them together randomly in fertilization, you increase the diversity further. And this number really only incorporates the ideas, or the concepts of independent assortment and random fertilization only. When you factor in the crossovers that occur during meiosis, then you greatly increase the genetic diversity possible. Well, it turns out that only a few traits are really encoded for by single genes. Most traits in the human body are going to be coded for by groups of genes that work together to determine those traits. When we're trying to figure out what the possible gene combinations are for offspring of parents with particular genotypes, we can use a simple diagram called a Punnett square to predict these gene combinations that are potential results of their mating. Let's look at Punnett squares using a relatively simple example, a single gene trait, the ability to roll the tongue or not to roll the tongue. That's actually an inherited trait, as it turns out. So as far as I know, most of us can roll our tongues, and if you can do that, you have an allele that would be the dominant allele, the tongue roller allele with a capital T. The recessive allele, lowercase t, can't roll the tongue. So if you're homozygous recessive, you have 2 little t's, and you wouldn't be rolling your tongue. But if you have a copy of the dominant allele, meaning you're homozygous dominant or heterozygous, then you're going to be a tongue roller because you have that dominant allele, and you only need 1 copy of the dominant allele to express that trait. So if we want to have 2 parents that are heterozygous, that is, they have this genotype, big T, little t, or dominant and recessive, they have a copy of each allele, and 2 of these parents, these 2 heterozygote parents mate, then what are the possible outcomes for their offspring? They've got both alleles. Turns out 75% chance of their offspring being a tongue roller. So the 25% chance of being homozygous dominant, 50% chance of being heterozygous. Either case, it's a tongue roller. And only a 25% chance of it being a non-tongue roller. And a Punnett square reveals this probability pretty easily. So we set up the possible gametes for each parent. In this case, in our example, each parent is a heterozygote. So the possible gametes from a heterozygote are one dominant and one recessive. And both parents are heterozygotes. So we have our female lined up on one side of the Punnett square, possible gametes from the female, and possible gametes from the male on the other side of the diagram. Again, he's a heterozygote as well, so one dominant gamete and one recessive gamete. Those are the possibilities. And then the Punnett square is going to reveal the possible offspring that result from those gamete combinations. So when these gametes come together, homozygous dominant. When this one and that one come together, contribution from each gamete leads to a heterozygote. Same thing over here, it's the same crossing. Dominant and recessive, you got a heterozygote. So there's 3 out of 4 ways to get to the tongue rolling phenotype. And when we combine these gametes, the possible combination is homozygote recessive. So there's a 1 in 4 chance of having a tongue roller. So that's just the probability of having a particular genotype in an offspring. But the more offspring you have, the more likely those genotypes are to actually follow the predictions of the Punnett square. So Mendel with his garden peas, where he was producing thousands of peas with each generation, he was able to see these probabilities in the number of offspring he had with different traits fairly easily. So the chance of having one child who's a non-tongue roller would be 25%. And when you want to consider the probability of 2 children being born as non-tongue rollers, you have to multiply the chances of each event occurring independently to come up with the overall probability. So it's a 1 in 4 chance times a 1 in 4 chance, or 25% times 25% chance. Multiply these together and it's a 1 in 16 chance, which is to say a 12.5% chance of having 2 children who are non-tongue rollers. The point is, each one is an independent event, independent of the previous child's genotype. So there's a fairly small chance of having 2 children who are homozygous recessive. Most traits that we can easily observe are actually encoded for by multiple genes. So like eye color, the example that I used previously in this lecture, that's actually a multiple gene trait. It's just an example that we use to illustrate some concepts. But there are multiple genes that determine eye color. And it turns out there's only a few traits in the body or the human that are actually dominant traits that are encoded by a single gene. So there are some examples like the widow's peaks and freckles and dimples that are actually dominant traits of single gene type traits. And diseases and disorders, inherited diseases generally are not dominant disorders, because that means the trait would always get expressed, and most of these disorders would be lethal. That is to say that they would kill the person who has that disorder before they get to the point where they're of reproductive age. So those traits generally aren't passed along very efficiently. Huntington's disease is an exception to that, because individuals with Huntington's generally are symptom-free until middle age. Sometimes recessive genes are going to confer the desirable trait, like normal vision is a recessive trait. There's an astigmatism where the lens or sometimes the cornea of the eye is curved differently between the right and the left eye. which leads to blurry vision. That's actually a dominant trait. So normal vision is recessive. That's not the norm, but some recessive traits confer the preferable condition. And remember, heterozygotes, which have a copy of both the dominant and the recessive allele, they're carrying the recessive trait. That is to say, they're not expressing it because they only have one copy. Being a heterozygote, they only have one copy of that recessive allele, so it doesn't get expressed because the dominant allele expresses instead. But they carry the trait, so they can pass it along to their offspring. Here's a list of some other traits in the body that are coded by a simple dominant recessive trait, like the tongue rolling thing or the astigmatism. And then there's an alternative to the dominant recessive inheritance known as incomplete dominance, where there's an intermediate phenotype that's displayed by the heterozygotes. So you actually have three phenotypes possible. One from the homozygote, homozygote dominant. Another phenotype possible from the homozygote recessive, and then a different phenotype possible with the heterozygote. So normally, both of these genotypes and the dominant recessive inheritance, both these genotypes would have the same phenotypes. In incomplete dominance, there's a third phenotype with the heterozygote. Sickle cell anemia is a good example of this, where homozygote recessive has the sickle cell anemia. And remember, sickle cell anemia is where you have a mutated hemoglobin. If an individual is heterozygote and they only have one copy of that sickle cell allele, then they have what's known as sickle cell trait rather than sickle cell anemia. So they can still suffer the same symptoms because they do have some mutated hemoglobin, but they suffer from these symptoms less frequently. Some genes come in more than two varieties. They have more than two alleles, even though we can only inherit two alleles at any given time because we have two copies of each gene. But there could be more than one allele that exists in nature. It's multiple allele inheritance. And the ABO blood group is a good example of multiple allele inheritance. I'm going to call these alleles A, B, or little I. A and B are codominant, which means if you have either one of these alleles, they are going to get expressed. Whereas little I is recessive, which means in order to express that trait, a person would have to be homozygous, little I. And that would be type O. So if you have A, you're going to be type A. If you have the B allele, you're going to be type B. If you have both, you'll be type AB. They are codominant. Either trait gets expressed if you have either of these alleles. And that way you can get 4 possible blood types, A, B, AB, or O, with just 3 different alleles. And in the case of the ABO blood groups, the homozygous recessive phenotype is the more frequently found in the population, whereas the codominant trait, where you have both dominant alleles expressed together, is relatively infrequent. Now, genes that are found on the sex chromosomes, the X and the Y sex chromosomes, have a different pattern of inheritance. These two chromosomes, sex chromosomes, are not equal to each other. The other chromosomes, the autosomes, the maternal and paternal copies of those chromosomes are pretty much identical. That is to say, they each have the same genes. But the X chromosome is much larger than the Y chromosome. So there are genes that are on the X chromosome that aren't found on the Y chromosome. The X chromosome has about 1,400 genes compared to about 200 on the Y chromosome. So those genes that you only find on the X chromosome are called X-linked genes. They're not found on the Y. So here's your X chromosome, and if you had 2 copies of these, you're a female. Here's the little guy next to it, the Y chromosome, much, much smaller than the X chromosome. So if you have X, Y, you're a male. And you can see there's much more room for genes over here than there is on the Y chromosome. So there's many more genes on the X than on the Y. So the consequences of that lead to sex-linked inheritance. When a male receives a X-linked recessive allele, it's going to get expressed. A male with an X-linked recessive allele only needs that one copy in order to express that trait, because he doesn't have another allele on the Y chromosome that could mask that recessive trait. Whereas a female, if she were to express the trait from a recessive allele on the X chromosome, she'd have to have both of those alleles on each of her on each of her X chromosomes. A couple of classic examples of sex-linked inheritance are hemophilia and color blindness, which are mostly expressed in the males because they only need the one recessive copy. But for the female to express that trait, she would have to have two copies of the recessive allele. So it's much more frequent in the males. So some traits follow the classic Mendelian patterns of inheritance, where there's essentially one gene that encodes for a trait, but many traits are going to be the result of different genes, multiple genes that together influence the same trait. Like eye color, the example I used earlier, the big B, little b. That's actually a polygene trait. There are multiple genes that contribute to eye color. Not just multiple alleles, there are multiple genes that together determine eye color, as well as height and, of course, many different genetic factors that play into intelligence and personality, etc. These are considered polygene traits. And they lead to continuous phenotypic variation. So height, for instance. Somebody's not just 5 foot or just 6 foot. There's a continuous variation in height, with individuals being of any particular height. Skin color is an example of polygene inheritance, where there's at least 3 different genes that play into skin color in an individual. A, B, and C, where the capital letters are going to be incompletely dominant over the lowercase recessive alleles. So somebody with all caps is going to be very dark skinned, homozygous. For all 3 of these genes, this would be a very dark skin. Homozygous recessive for all 3 of these genes would be very light skinned. So if you, for example, took an individual who's very dark, crossed them with a very light skinned individual, and the offspring are going to be all heterozygous, where the pigmentation is going to be somewhere in between. And then if you can continue this experiment for another generation, you took this first generation of heterozygotes that resulted from this cross, and you cross them back with each other, your second generation offspring would have even a greater variation in the phenotype. And phenotypic variation in a polygene inherited trait is going to exhibit a bell-shaped curve. There's a lot of individuals who represent an intermediate type phenotype, and fewer that are at the extremes of the phenotypic variation. So your very light skinned and your very dark skinned individuals, there would be fewer of those, and there would be more in between that show some sort of incomplete, or I should say a intermediate phenotype. Phenotypes, as it turns out, are not just dependent on the alleles that are inherited alone. There are environmental factors that influence gene expression as well, and we're really just beginning to understand this. For instance, many drugs can influence gene expression patterns, and this is particularly important while the mother is pregnant with the baby. Things that she takes could potentially influence the expression of genes in that developing fetus. Thalidomide is a classic example. This is a drug that used to be prescribed to help counter morning sickness in pregnant females, but it turns out that this led to horrible birth defects, and so it was pulled from the market eventually. This is an example of a phenocopy, where a drug produced an led to an environmentally produced phenotype that mimics a phenotype that could be seen with genetic mutation. And other environmental factors can influence gene expression after birth. Nutrition plays an important role, of course, in development. So having inadequate nutrition is going to lead to particular phenotypes. And also childhood hormone abnormalities can lead to differential growth. So this is going to also lead to particular type of phenotypes that go beyond just the genetic factors. Genetic screening can be used to detect inherited disorders, and it's generally used to routinely screen for fairly common genetic disorders. And sometimes parents will want to know if their child has a chance of inheriting a trait that they might have, such as Huntington's disease, or maybe a woman who is of a somewhat advanced age might want to know if her child has trisomy 21, which leads to Down syndrome. Women of advanced age have a greater chance of missegregating this chromosome number 21. So these are examples of things that somebody might screen for. This can be done using pedigrees. When you track a trait through different generations, following their phenotypes, and then one can kind of deduce by working backwards genotypes that people have based on the phenotypes that are exhibited in the pedigree. So I'm not going to go into this how you actually do this, but here's an example of a pedigree showing generations of people who have either the widow's peak or don't have the widow's peak, where the widow's peak is in purple, unaffected folks are clear, males have squares, females have circles, and here's a mating between widow's peak phenotype and a non-widow's peak. And from their number of offspring, you follow this through enough generations and you can start to deduce the genotypes. That's one way to do genetic screening. Blood tests and fetal testing are other ways to do more definitive genetic screening. So fetal testing isn't used unless there's an actual concern, because it's an invasive technique and there's a risk, obviously, to both the mother and the fetus. A couple of different types of fetal testing: amniocentesis and chorionic villus sampling. So in amniocentesis, they actually stick a needle up in there and withdraw some of the amniotic fluid. And in chorionic villus sampling, what they do is they enter through the cervix with a suction device, and they take off some of this chorionic villi, which is part of the placenta. And then they can go and take the chromosomes from that. They stain the chromosomes with some fluorescent dyes, produce a karyotype where they can look at the chromosomes and check it for abnormalities. The chorionic villus sampling can be done earlier, 8th to 10th week of pregnancy versus 14th to 16th for the amniocentesis. And also the lab work for the chorionic villus is done more quickly than the amniocentesis. But they both give information about chromosome abnormalities like translocations and things like that. So currently, field testing, blood testing, and all that is being done just to detect genes and the potential for traits. But one day we'll potentially be able to genetically engineer some of those undesirable or defective genes out of individuals and replace them with the safer, more desirable gene. This has long had a controversial past for various reasons, but we've made advances in genetic engineering, such as CRISPR technology. You may have heard of CRISPR. CRISPR is a new genome editing technique that holds a lot of promise, that offers the potential to actually splice out a defective gene and replace it with a desirable copy of the gene. So stay tuned for that.